The overall goal of this procedure is to identify co-evolving residues in protein alignments that imply interpositional dependencies, or IPDs. This is accomplished by first loading the alignment into StickWRLD, which is a visual analytics tool that creates an interactive 3D representation of a protein alignment and clearly displays covarying residues. In StickWRLD, each position in the alignment is represented as a column comprised of a stack of spheres, one sphere for each of the possible 20 amino acids in that position within the alignment that are sized in a frequency-dependent manner. The columns representing each position are wrapped around a cylinder. To represent IPDs, lines are drawn between residues which are co-evolving higher or lower than would be expected if the residues present in the positions were independent. Within the program, the residual is tuned until there are a manageable number of edges, and then the edges of interest are identified. Ultimately, StickWRLD is used to identify residues which are co-evolving with one another. Such functionally required covarying residues have been identified in adenylate kinase.
One of the reasons people struggle with this process is because of the novelty. It's almost as much of an art form as it is a science.
Demonstrating StickWRLD visually is important because it's a visual analytics tool and requires user interaction.
Use a computer that has an Intel i5 or better processor with at least 4 GB of RAM, is running Mac OS X or Linux OS, and is equipped with the Python libraries listed in the text protocol. Download StickWRLD as a zip archive containing all of the relevant Python scripts. Also download the FASTA